So it's a great pleasure for us to introduce you that are a professor of medicine and director of basic liver research at Cedars-Sinai Medical Center in Los Angeles. And he also holds professorship at the David Geffen School of Medicine at UCLA and at Charles Drew University College of Medicine. Dr. Seki trained as a hepatobiliary surgeon in Japan and later conducted postdoctoral research at the Columbia University under Dr. David Brenner. He's internationally recognized for his pioneering study on the gut-liver axis and tool-like receptor signaling in liver disease. At Cedars-Sinai, his laboratory has made significant contribution to understanding liver fibrosis, including the identification of hyaluronan as a fibrosis biomarker and therapeutic target, with landmark publication in Nature Medicine, Science Translational Medicine, and multiple other journals. And Dr. Seki has already published more than 150 peer-reviewed papers, cited more than 40,000 times, and has received multiple awards from the AASLD and the American Liver Foundation. So Dr. Seki, thank you very much for joining us today, and we look forward to hear from you. Thank you.
So, hi everyone. Thank you for giving me an opportunity to share my recent research work through this yellow webinar series. I'm Ekihiro Seki, Professor of Medicine, Cedars-Sinai Medical Center, Los Angeles, USA. So this is the outline of today's my talk. First, I will introduce very briefly about metabolic dysfunction associated steatotic liver disease, MASLD, formerly known as NAFLD. Then I will discuss fatty liver, colorectal cancer, and liver metastasis. Then moving forward to mechanism, we will discuss about fatty liver effect on metastatic tumor growth. Then we'll discuss about the effect of tumors on fatty liver and fibrosis. Then finally, I will discuss about fatty liver effect on immune microenvironment in liver metastasis. So in the United States, 50 to 60% of adults are obese, and 25 to 30% of adults have MASLD. This is currently also similar in the other area in the world. Like India, we know 40 to 50% people have MASLD. Here you can see many lipids are accumulated in hepatocytes. Why this condition is bad? Because its complication is bad. MASLD can develop to MASH and further fibrosis and cirrhosis, which further causes esophageal varices, ascites, and liver failure. Those are all life-threatening complications. Also, we know fatty liver is the risk factor for hepatocellular carcinoma, HCC, also associated with cardiovascular diseases. Recently, people pay more attention in extrahepatic cancers in fatty liver patients, such as colorectal cancer, gastric cancer, lung cancer, breast cancer, prostate cancer, and pancreatic cancer. MASLD is a spectrum disease starting from hepatic steatosis and developing to MASH, which is steatosis with inflammation and fibrosis, and then cirrhosis. Also, some of them will develop to hepatocellular carcinoma, HCC. Interestingly, MASLD HCC can be developed any stage of MASLD. So MASLD is a significant risk factor for HCC. So this is a data from New England Journal of Medicine 20 years ago. This data shows higher BMI is associated not only with primary liver cancer, but also with other cancers as well, such as prostate cancer, colorectal cancer, breast cancer, and pancreatic cancer. Also, a few years ago, NCI, National Cancer Institute, also highlighted higher BMI is a risk factor for colorectal cancer. So this study from Mayo Clinic followed up 19,000 patients for 21 years. And this paper actually compared obese people without fatty liver and with fatty liver. Very interestingly, obesity alone did not increase the risk of cancers. So these blue labeling, but you can see this red dot. Fatty liver patients significantly increase cancer incidence, such as liver cancer, colon cancer, pancreatic cancer, gastric cancer, and breast cancer. So I will move to the more about fatty liver and liver metastasis. So, as you know, all cancers have a potential to metastasize to the liver. Colorectal cancer, pancreatic cancer, gastric cancer, breast cancer, melanoma, and lung cancer. Because of unique anatomical link between the GI tract and the liver, GI cancers often metastasize to the liver. With treatment, 5-year survival of colorectal cancer is now around 50 to 60%, pretty good. However, 50% of colorectal cancer patients develop liver metastasis. And in those patients, 5-year survival, unfortunately, down to 13%. So we can tell liver metastasis is a survival determinant for colorectal cancer patients. Also, 80% of patients with pancreatic cancer, much worse, will develop liver metastasis, and their 5-year survival is almost zero. The prognosis of patients with liver metastasis is very poor, but their survival rate, recurrence rate, and response rate to anti-cancer treatment are distinct among patients. Why? Of course, cancer characteristics are different in each patient. Tumor heterogeneity should be very important. What about force factor? For example, heterogeneous patient population, such as patients with fatty liver. Since 30% of adults have fatty liver, we should consider this condition. This data demonstrated colorectal cancer liver metastasis is affected by fatty liver. This report from United Kingdom studying 1,800 patients. This paper showed after liver tumor resection of liver metastasis, the recurrence rate of liver metastasis increased and survival rate is decreased in fatty liver patients compared with non-fatty liver patients. In other study also showed they investigated 3,200 patients, and this study also supported muscle D worsens the prognosis of colorectal cancer. So this is another study from Harvard University, United States. This study also supported fatty liver reduce the survival of colorectal cancer patient. So this is a historical picture. One day, one of the pathologists from our institution actually came to me and showed this slide. So these areas liver metastasis. This is the liver, and these are non-tumor liver area. You can see here a lot of the fat deposition. So he found many liver metastasis patients in other field also showed fatty liver. And in our cohort actually showed 45% of patients have this type of fatty liver condition. Thinking about generally rate of fatty liver is 25%. So our cohort here, I show the table of our cohort. 34 patients, actually very small. However, compared with the general population, thinking about 25% of the fatty liver patient, so our cohort showed 45%. So pretty liver patient showed pretty often fatty liver. So this is another study from China. They analyze synchronous liver metastasis. They say when patient came to the doctor, oh, so this synchronized liver metastasis is when patient came to the doctor, these patients already have liver metastasis. So liver on the left panel, this data actually showed, looking at stage 4, I highlighted, these are more, these stage 4 patient have more fatty liver patient. And also you can see the right panel. This showed NAFLD has correlation with liver metastasis in addition to tumor marker CEA and CA19-9. This study also analyzed the relationship with FIB4. Higher FIB4, more fibrosis, showed less metastasis. This could be due to reduced portal vein flow by cirrhosis. This is consistent with all the studies showing cirrhosis patients are less metastasis. However, recent studies also showed more mixed observations. Some studies showed even with cirrhosis, metastasis rate can increase. We need more investigations. So here, we hypothesis fatty liver may have different liver microenvironment, which may explain the heterogeneity of metastasis rate, growth, and the response to anti-cancer therapy on patients with liver metastasis. If mechanisms are different between the patients with and without fatty liver, different disease management should be considered between the patient with and without fatty liver. So this paper demonstrated fatty liver can affect liver immunity and reduce response to immune checkpoint therapies. So this is a very famous study from Heikenwalder's group published in Nature a few years ago. This study shows patients and mice with massive HCC are resistant to anti-PD-1 therapy. HCC even got worse. In these cancers, they observed CD8 T cells are increased, but these CD8 T cells highly express PD-1, indicating CD8 T cells are exhausted, and these cells cannot kill cancers anymore. Also, this study is published from Derek Mudd's group in the UK. This paper showed very similar results with Heikenwalder's group. So HCC mice with Western diet are resistant to anti-PD-1 therapy. But when they inhibited CXCR2, indicating inhibition of neutrophils, they recovered efficacy of the anti-PD-1 therapy, demonstrating the possibility of modulation of a tumor microenvironment can improve the response to immune checkpoint therapy in MASLD HCC. Based on these papers, we also hypothesized a similar condition in fatty liver-related liver metastasis. Fatty liver could affect immune microenvironment and response to immune checkpoint therapy for colorectal cancer liver metastasis. Then we will move to the, some discussion of the mechanisms. So to understand the mechanism, we use animal model. To develop liver metastasis, we use splenic injection model. As you can see the right panel, splenic injection, splenic vein merges to the portal vein. This model can take the same metastasis route with natural liver metastasis of the colon cancer through the portal vein. So we believe this approach can model colorectal cancer liver metastasis mechanism very nicely. So here, I want to introduce a bit about extracellular vesicles. EV as a mechanism. So EV can be produced from any cells. And there are different sizes of EVs. So exomer, exosome, microvesicles, large oncosomes, and apoptotic bodies. So in our this study, we mostly discuss about exosome in our study. So as you can see, extracellular vesicles, EV contains many bioactive molecules such as proteins, lipids, DNA, and RNA, and transfer these bioactive molecules from one cell to other cells. Here, we want to discuss from hepatocytes to the cancer cells. Then we wanted to know the mechanism, how fatty liver enhance tumor growth. We focus on extracellular vesicles EV. As you can see, blood EV number is increased in muscle D patient. My former postdoc, Dr. Wang, was interested in the effect of EVs from muscle D to the cancer cells. He extracted EVs from blood from patients and mice with fatty liver. He also isolated EVs from culture media of primary hepatocytes treated with palmitate, creating fatty liver hepatocytes in culture. Then he put these EVs on cancer cells. And he found these EVs can enhance cancer cell proliferation, migration, and invasion, demonstrating EVs can enhance tumor growth in vitro. So our next question is, what is in vivo? He blocked EV production in hepatocytes by silencing YW2025 in hepatocytes. After the establishment of the fatty liver by 6 weeks of high-fat diet feeding, YW2025 is a critical factor to produce EVs from cells. So silencing YW2025 should reduce EV levels in blood, which is increased by high-fat diet. Expectedly, so you can see inhibition of EV production inhibited tumor growth only in the high-fat diet condition, suggesting fatty liver-derived EVs are important for tumor growth in fatty liver. So right panel showed NCI kindly highlighted our paper in their website saying fatty liver sends message bubble to cancer cells to grow and enhance metastasis. So we also overexpressed YW2025, which increases EV production. And also we injected EVs from fat accumulated hepatocytes, both YW2025 overexpression and EV injection increase liver metastasis. So next question is, what factors in EVs are important? We analyze microRNA profile in EVs from patient and mice with fatty liver, and also we extracted EV from in vitro fatty liver hepatocytes treated with palmitate. We found 6 microRNAs are commonly upregulated in these 4 different cohorts. Among 6 microRNA, we found 3 microRNAs commonly inhibit one molecule called LATS2, which is a negative regulator of YAP signaling. So increase these microRNA can suppress LATS2 level, then activate YAP activity to enhance tumor activity, including metastasis. So what is YAP? YAP is a transcription cofactor for pro-cancerous TID transcription factor and the core element of the HIP-YAP signaling pathway. So YAP signaling is important for development, tissue regeneration, and cancer activity, including proliferation, migration, invasion, EMT, and metastasis. So our data showed exactly these 3 microRNA can activate YAP. So you can see here, YAP nuclear translocation in the cancer cell by treating with these 3 microRNA. We then checked YAP pathway in fatty liver metastasis model. In fatty liver metastasis, our RNA sequence data showed YAP signature and cancer-related signature are enriched, indicating metastatic tumors are more aggressive in fatty liver, likely by YAP. Then, right panel shows when we knock down YAP in cancer cells, expectedly, tumor formation was decreased, showing YAP is important for liver metastasis enhanced by fatty liver. Then we moved to the next topic. So the effect conversely, we wanted not to know the effect of the tumors to the tumor microenvironment, such as here is fatty liver and also the fibrosis. So I want to highlight these 2 papers from Dr. Levitt Leiden from Weill Cornell University, New York, US. Dr. Leiden is a pioneer to establish the concept of pre-metastatic niche formation. So this concept is cancer can create tumor-permissive microenvironment before cancers physically come to the metastasis organ here in the liver. So here is cancer-derived EV play a role in this mechanism. So you can see that in the left paper, he showed pancreatic cancers produce EVs. This EV first come to the liver before cancer come. EV content MIF, one of the chemokine, this stimulates Kupffer cells and hepatic stellar cells to produce TGF-beta and fibronectin to create tumor-permissive microenvironment in the liver, and then liver metastasis occur. The right panel showed cancers produce EVs. These actually come to the liver and develop fatty liver through this EV content, palmitate, and also the TNF. And that palmitate and that stimulate TLR4 and TNF in hepatocytes to create more fatty liver condition that affect the tumor growth in the liver. So now we are more interested in about fibrosis in cancer. So we previously reported extracellular matrix hyaluronic acids play a significant role in developing liver fibrosis by activating hepatic stellar cells. This study was done by my former postdoc, Dr. Yunmi Yang. Currently, she is an associate professor at Sunkyung Kwon University in the South Korea. We found enzyme HAS2 expressed in the hepatic stellar cells is responsible for hyaluronic acid production, stellar cell activation, and fibrosis. So Dr. Yang continued the study and tried to answer the question about the role of liver metastasis and fatty liver in fibrotic tumor microenvironment. Colorectal cancer is very heterogeneous. Some patients have fibrosis in tumors, the others do not. We used MC38 syngeneic colorectal cancer cell line for liver metastasis model, and you can see in low-fat diet condition, this cancer metastasis model this is cancer area, do not show fibrosis in tumors. However, when we fed high-fat diet in these animals, no fibrosis is observed in non-tumor region, but we observed in the tumors a lot of fibrosis. So collagen deposition in the here, and also hyaluronic acid deposition. And in these fibrotic tumors, we concluded HAS2 and hyaluronic acid play significant role in the tumor development. You can see hepatic stellar cell spike HAS2 knockout mice showed reduced liver metastasis, which is actually increased by the high-fat diet feeding. So this is showed decreased. So we finally found a series of experiments, and we found tumor-derived CTGF through YAP activation in the cancer cells play a significant role in the stellar cell activation and it has to upregulation. So this has to upregulate stellar cell further produce hyaluronic acid through CD4 activated YAP in the cancer cells. So we concluded there is a feed-forward loop between the tumors and the stellar cells. And the tumors promote stellar cell activation and has to upregulation, which further enhance tumor growth through the CD44 and YAP. So final question is how fatty liver affect immune microenvironment in liver metastasis. So now we are interested in tumor-associated macrophages, especially the role of M2 macrophages. So as you know, M1 is anti-cancer. Conversely, M2 is pro-cancer. And M2 macrophages can be classified to the M2A, M2B, M2C, and M2D. So M2A is more like these macrophage has phagocytic capacity. M2B is immunomodulatory. These are M2 macrophage, however, express IL-1 and IL-6. M2Cs, we can tell this fibrogenic macrophage. They produce TGF-beta and express MARTK. M2D is more angiogenic tumor-associated macrophages. They express angiogenic factor, VEGF, TGF, and also anti-inflammatory factor, IL-10. So you can see the right panel. We found this high-fat diet condition increase tumor-associated macrophage infiltration. In here, you can see, compared with low-fat diet. And in these tumor-associated macrophages is more like M2 dominant. You can see expressing on the CD206. And when we actually knock down YAP in the cancer cells, so we knock down cancer cells, but we found M2 tumor-associated macrophage infiltration is decreased. And we suggested this YAP-mediated M2 tumor-associated macrophage could promote metastatic tumor growth. So what is mechanism? How YAP contribute to the M2 TAM recruitment? So you can see here, we focus on the one of the downstream molecule called CYR61. This is actually encoded by the one with the gene called CCN1. You can see high-fat diet feeding increase CYR61 expression here. So this is immunohistochemistry, but this expression is actually decreased by silencing YAP in the cancer cells. So we think cancer YAP can lead to the induce one with the chemokine, so CYR61, to increase tumor-associated macrophage recruitment. So expectedly, when we knock down CYR61 here, CCN1, in the cancer cells, increase M2 macrophage infiltration is decreased. And also we found cancer growth in this CCN1 silenced tumors. You can see less tumor progression. But this is we only express in the condition of the fatty liver. So only in the fatty liver condition, this CCN1 production, upregulation from cancer cells through the YAP actually enhance the cancer, not in the low-fat diet condition. So then we are really interested in more mechanism related to the immune microenvironment. So our lab scientist, Dr. So-Young Kim, analyzed single-cell RNA sequencing. So we found M2 macrophages. So this a lot of clusters. So we first analyzed M2 macrophages. So we found particularly M2B like macrophage subpopulation was really increased in the fatty liver. And those M2B macrophages is highly expressed tumor promoting factors such as VEGF, TGF-beta, and IL-1 beta. we also found these macrophages also enhance expression of immune checkpoint molecule, such as PD-L1 and TIM3. Interestingly, we found similar to MASH HCC, CD8 T cells were enriched in fatty liver tumors, and these CD8 T cells highly express PD-1. And also not only the PD-1, other immune checkpoint molecules such as PD-L1, PD-L2, TIM3, CTLA4, LAG3 are highly expressed, indicating these CD8 T cells are exhausted and induce immune microenvironment. These actually conditions, although CD8 T cell has anti-cancer activity, these exhausted CD8 T cells cannot kill cancer. And also this condition could be the mechanism of the resistance to immune checkpoint inhibitors. So this diagram, recently we published one review paper in the trend of the endocrinology and metabolism. So we picked one diagram from this review article. So this diagram tells various immune cells contribute to the immunosuppressed microenvironment in fatty liver. So not only PD-1 positive CD8 T cells, but also neutrophil, myeloid-derived suppressor cells, M2 tumor-associated macrophages that can produce PDGF, TGF-beta, and IL-1. And also, we believe hepatic stellate cells contribute to the immunosuppressive microenvironment in fatty liver. So right panel shows the response to anti-PD-1 antibody is very effective in the low-fat diet condition, but less effective in high-fat diet condition due to very high expression of immune checkpoint molecules in this high-fat diet condition I showed in the previous picture here by the single-cell RNA sequencing. So we wanted to translate our mouse data to human study using clinical samples. So we created a tissue microarray here containing primary tumors, liver metastasis tissues, and also non-tumor liver tissues from patient with and without fatty liver. So using this tissue microRNA, we can analyze this immune microenvironment at once. So what we performed multiplex immunostaining using 40 different antibody, which are conjugated with different metal conjugated antibody, and using CyTOF technology and imaging mass cytometry technology, we scanned slide and captured signal and computationally analyzed those signals at single cell levels. We analyzed a total 430,000 cells from 30 patients. So we analyze, here you can see this UMAP picture. So we analyze immune cells and the tumor cells. And in muscle patient, you can see more M2 macrophages and CD8 T cells in tumors. And also we found these M2 and CD8 T cells more immune checkpoint molecules such as PD-1, PD-L1, and VISTA, and TIM3. And also I can tell this analysis can preserve spatial information, so location information. So we analyze distance between the cells. So here we analyze YAP expressing tumors and YAP negative tumors and distance with M2 macrophage and CD8 T cells. And you can see M2 macrophages are really closer to the YAP cancer cells compared with YAP negative cancers. In contrast, CD8 T cells are far from YAP cancers compared to YAP negative cancers, indicating so YAP expression is contribute to the M2 macrophage recruitment. Likely this is through the YAP mediated CYR61 expression. And the exhausted PD1 expressed CD8 T cells is very difficult to be recruited by the YAP positive tumors. This actually we can explain how M2 macrophages, CD8 T cells contribute to the tumor expression, tumor progression. So we further analyze the relationship between the cancer-associated fibroblasts and macrophages. So here we compared CD44 positive and negative macrophages because we are interested in study hyaluronic acid HA, and CD44 is a receptor for the hyaluronic acid HA. So here you can see CAF density. We compare the CAF density. CAF density is higher or long distance between the CAF and the M2. So you can see the higher density the CAF or closer distance between the CAF and M2. CD44 positive T cells here and here. So CD44 positive macrophages, M2 macrophages express higher PDL1. These data demonstrated YAP, CD44, and CAF can regulate immunosuppressive microenvironment through PD1 and PDL1 signaling. We also analyzed primary cancers. I mentioned our tumor tissue microarray contain primary tumor and metastatic tumor tissues. So here you see the Ki67, which is a proliferation marker, and YAP, we think this is aggressive marker for the cancers. Expectedly, liver metastasis in muscle D patient here had higher Ki67 and YAP expression compared with non-fatty liver patient. So to be surprised, this is surprise. Primary cancers also had higher Ki67 and YAP expression compared with non-fatty liver patient. So this data suggests muscle D affects primary cancers to make cancers more aggressive, and this can promote more liver metastasis in the muscle D patient. So we showed liver metastasis with fatty liver is resistant to anti-PD-1 therapy. So here, low-fat diet showed more effective in anti-PD-1 therapy, but high-fat diet condition is not really effective. So my current postdoc, Dr. Ji-Yoon Kim, actually wanted to address how this less effective of anti-PD-1 therapy in the muscle D, how we can improve about this. So she actually suppressed hyaluronic acid production by 4-MU here. And she found the efficacy of anti-PD-1 therapy is actually recovered here. This data demonstrated modulation of tumor microenvironment here, hyaluronic acid mediated fibrosis could improve the effectiveness of anti-PD-1 therapy in fatty liver conditions. So for fatty liver patients, we may have to consider different anti-cancer management. So this is a summary of our talk. First, I discussed hepatic steatosis is a risk factor for colorectal cancer as well as colorectal cancer liver metastasis. And then we discuss muscle D-derived EVs promote metastasis to the liver via their oncogenic cargo microRNAs. And also we discuss muscle D modulates tumor microenvironment to enhance extracellular matrix production, especially collagen and hyaluronic acid, which further promote tumor growth. Also, we discuss cancer YAP activation is crucial for creating immunosuppressive tumor microenvironment in muscle D. And also we discuss muscle D can limit anti-PD-1 treatment for colorectal cancer liver metastasis, and modulation of the tumor microenvironment may improve the efficacy of the anti-PD-1 therapy in the muscle D patient. So this is my very important message from our study. We may have to consider different disease management for patient with colorectal cancer or colorectal cancer liver metastasis with or without muscle D. So this study actually started from Dr. Yu Mian and were followed by the Dr. Wang and Dr. Kim, especially Dr. Soyeon Kim is all worked for our single cell analysis, and Dr. Ji-Yoon Kim worked on our anti-PD-1 therapy. So I want to really, thank our collaborator at Cedars-Sinai and other institution, and also our former lab member and funding from NIH. Thank you for your attention.